Today, we're going to run an experiment together to see how different treatments change the time it takes for seeds to germinate. For more details on how germination works, be sure to check out the Botany Besties website. In your kit will be a little egg crate and a bag of soil and a legend. It's going to look like this. This is going to tell you which seeds we're going to plant in which cells. The first thing that you're going to do is take your egg crate and fill up each of the 12 cells with some of the soil. This soil is a soilless medium. It's the Jiffy Seed Starter. Next, we're going to get started planting. So the lupine seeds that we're going to plant first are the dry lupine seeds. So we'll plant these in the corresponding cells. So there should be 3 seeds for you to plant in the cell. These seeds do need to be covered a little bit, just so they can't see the light. So you can just give them a little tiny poke in, not think too much, and just brush the the soil over them to make sure that you can't see the seeds. Next, we're going to plant the lupine seeds that are imbibed. When a seed is imbibed, it means that it has already swollen up with water. So it means it has completed the first stage of germination. These seeds, when we imbibe them, it helps them start germinating more quickly, just by soaking them in a cup of warm water overnight. We're going to plant 3 of these seeds in the lupine imbibed cell. And then we're going to poke them in just like we did with the other ones, and brush the soil over them to make sure that they can't see the light. Because sometimes plants won't germinate if they can't see the light, or if they can see the light in this case. Our next section is Asclepias syriaca seeds. Asclepias syriaca is also known as common milkweed. So we're going to take 5 of these seeds, which we collected during the summer right after the seeds had released, and we're going to drop these onto the surface of our soil. We do want to make sure that they're a little bit spread out so we can tell when each of the plants sprouts. And just like the lupine seeds, we're going to poke them in a little bit and just to anchor them, and then brush the soil over them so they can't see the light. These seeds actually won't germinate if they can see light. So this step is important, but if you poke them in too far, they're going to have trouble getting up to the light before their leaves start photosynthesizing, so they might have trouble germinating. So next we have this section that says Asclepias syriaca moist stratify. So moist stratify just means that, for example, you take the pods of the Asclepias and you give them a little bit of water, so you moisten them up, and then you put them in a baggie and you put them in a fridge for anywhere from 2 weeks to 3 months. We actually took these from outside during the winter after it had snowed, and then added a little bit of paper towel into the baggie and made sure that was moist along with the seeds. So now we're going to take 5 of these seeds and put them into the cell. You can pull off the fur if you want. 1, 2, 3, 4, 5. And just like we did with the others, we're going to give them just a little bit of a poke in, and then we're going to fluff the dirt right on top of them. It's some Asclepias first time. Alright, so the next seeds that we're going to plant is all of the carrot seeds. These are all the same seeds. We're just going to water them for the initial watering with a different solution. So these are just Carota carrots. They're a garden variety of carrots. And you're going to take the seeds in this baggie, and you're going to put 5 of the seeds in each of these cells. So I already put 5 in each, and these ones you are not going to bury. You just want to let them rest on top of the soil. The carrots don't actually like to be buried. They can germinate in the dark, so that's not actually the problem. But they're just really delicate when they sprout, so it's way too hard for them to get up through the top of the soil if they get buried. So in your kit, we had treatment waters. So these treatment waters are in jars, jam jars, and on the top you can see they'll say the treatment name, like this one says xylitol. So this one we'd plant down here. So since this is the xylitol one, we're probably going to actually wait on that and try and go across. So we're going to start with the baking soda jar. And we're going to take a syringe and measure out 5 milliliters of this solution. When you measure the 5 milliliters, you have to make sure that the bottom side of your plunger is at the 5 milliliter mark. So just make sure that there's 5 milliliters of water in the open part of the syringe that has the water in it. So it's at the very bottom side of this plunger. And we're going to take that and put it on top of our carrot seeds in the cell. If it's not draining too well, that's fine. It is the first time that this soil has been watered. So just be careful. So now we're going to go to the next treatment, and that's with vinegar. So the baking soda and the vinegar and the next two are known to speed up carrot seed germination, but we don't know how much. So we're going to try and see if any of these speed up the germination more than the others. Now make sure that you use either a different syringe or you rinse out your syringe between treatments, because you want to make sure that there's no cross-contamination between your different ideas, because that would make sure, or that would combine them, and that might tell you that vinegar and baking soda together don't have an effect off of the control. That would be because vinegar and baking soda, one's an acid and one's a base, so when combined they make water. So we're going to use a separate syringe. I'm just going to draw 5 milliliters of the solution, and I'm again making sure that the bottom of my plunger is at the 5 milliliter mark. And then I'm going to add that into the next cell that corresponds to vinegar. It's okay if it flows over a little bit. We're just trying to be as accurate with this as we can. Actually, I think we might have a little extra there. We got a little long carrot seed that I'm going to pull out. Alright. So our next treatment is hydrogen peroxide. And I'm going to drop, again, 5 milliliters of this solution. And I'm going to water this cell with that. My legend's getting a little wet here. There we go. Alright. And next we're going to add the aspirin solution. So this aspirin solution was pre-mixed, so it's actually extremely dilute. This is about 300 milligrams of aspirin in a liter of water. So a liter is about a quarter of a gallon or so. And we're gonna just draw this up again with 5 milliliters. So these carrots are getting very little amount of aspirin, but it actually had quite a drastic effect on decreasing the time it took for the carrot seeds to germinate. Alright, and so we're going to take a quick break here because I have to go rinse my syringes, and when we get back we're going to finish the last 4 treatments. And then we're going to talk about what a control is in an experiment and how to use it. Hello everyone! So we're back, and I just rinsed out my 4 syringes. So these next 4 cells, or 3 of them, are a complete experiment. With the sugar water and the Epsom salts and the xylitol water, we have no idea what's going to happen. It's never been studied. So we're going to have fun finding out together. So we're going to do the carrots with sugar water first. And your lid should say sugar on it. And drop 5 milliliters of the solution into the bottom of the plunger. And then add it to the sugar water cell. Next, we're going to do carrots with Epsom salts. So Epsom salts are also another type of salt, but instead of being like normal salt, which is made with sodium, this is made with magnesium. It's actually used for medicinal purposes, generally. It decreases swelling. But for plants, it actually, well, what we think might happen, this is our hypothesis, it's going to increase salinity of the soil. And that will definitely affect germination, or so we think. But we're not sure which way. So we're going to find out. We'll get a fresh syringe. And drop 5 milliliters of the Epsom salt water. So now we're going to put it into our corresponding cell. And now we're going to skip a cell and go to the xylitol cell, which is right here. So xylitol is a sugar substitute. To our mouths, it tastes like sugar, but it actually isn't anything like sugar. It's a completely different group of compounds, so our bodies don't recognize it as sugar. And plants, for this reason, might not recognize it as sugar either, because sugar is a fuel for them, just like it is for us. So we're going to see what happens when we add the xylitol. I'm actually really curious to know how this is going to turn out. So again, I'm drawing up 5 milliliters. And I'm going to put that in. And if it bubbles up like this, that's fine. This dirt has never been watered before. But eventually the water will sink in. You just gotta be a little careful not to spill. And now this last cell, it says carrots and then parentheses control. So a control is one treatment that is your baseline treatment. So what's going to happen if you don't treat it with anything. In our case, we're treating all these other carrot seeds with like baking soda, vinegar, aspirin, Epsom salts. Each of those is going to be another treatment, so we're adding something to the water. So for our control, we're not going to add anything to the water. We're just going to give them plain water. So we have in our applesauce jar here some plain water. On the cover you'll see it says reverse osmosis, or R osmosis, water. So that's a fancy way of saying that this water is very well filtered. So there's going to be only a few like salts or other impurities, but it's mostly just going to be water. And we're going to take 5 milliliters of this water, or this control water as well, and we're going to put it into our control group. And now we've planted all of our seeds. From now on, what you're going to do is you're going to make sure that these stay wet. One way you can do this is by watering them, like pouring a little bit in with a syringe of this plain water, but you gotta make sure it's plain water. We've already done the initial treatment, and now to keep them wet, we're just gonna add the plain water. The other way that you can make sure that they're wet is by using a spray bottle. And this is one of the most easy methods. So you're just going to take your egg crate, and every day you can look at it, and if it's like super dark, almost black, and a little bit shiny, then you don't need to add any water. But it's likely that for the first few days at least, you're going to need to spray your egg crate down. And you spray it until the surface looks black and shiny all over. And then you can wait until the next day to water it again. The other reason that you're checking on this every day, other than making sure that it's moist, is to make sure that you know when your plants germinate. You're just going to look for whenever the little seed head or the seed starts to crack open and a little seedling starts to stick up out of your egg crate. And then you're going to mark down the date that that happens on whichever one of the cells that it happened in. For example, if like 3 days from now, the lupine cell germinated, then I would write January 30th, first germination on my lupine cell, whichever one it was. If it was dry, I'd be a little surprised, but I'd write that down. So we want to know when the first seed germinates. And from there, you can also write down like when the last seed germinates, or if all of them germinate, because maybe all of them don't germinate, or maybe none of them germinate. Maybe one of our treatments, like the Epsom salts for example, if only 2 of the seeds germinate in there, but 4 or 5 germinate in the rest of the cells, maybe the Epsom salts was actually decreasing the number of seeds that could germinate. So we're going to write down when the first seed germinates and how many germinate at the very end. And then we're going to compare our data and send it all together and see how the treatments affected the time that it took for each of the seeds to germinate. Once your seeds are planted, you're going to need to make sure that they stay moist at all times. And one of the ways that you're going to do this is you're going to make sure that the top on your egg crate stays closed. And this is fine because most seeds need light to germinate, but these ones do not. So if we close the top in their egg crate, then it's just going to serve to keep them moist while they're germinating. This will also help them imbibe because there will be more water in their surroundings. Every day, you need to check, for one, is it germinating? And then write down if it is, like we described before. But you also need to check if there's enough water. And the reason that you'll know when you need to water is if there's any signs at all of a lighter brown patch or little light brown speckles on your soil, or anything other than a dark brown uniform piece of soil, then you need to water. And the easiest way to do this is with a spray bottle. So you're just going to mist your egg crate until it appears to be dark black and shiny, or almost black. And then you're going to just let it sit until the next day, and then again, check if it's germinating, and check if you need to water it. After a few days, it's possible that your soil is going to stay the nice dark black with a little hint of shiny that we want. And this is because the Jiffy Seed Starter has a moistening agent in it. So that's going to make sure that once the soil gets moistened fully, it's going to stay moist. And then after your seeds start germinating, don't forget to leave your top open, because all seeds, once they germinate and they start growing the little seedling tops, they need to have light. This is how we make sure that the plants can perform photosynthesis, which is how they make their food so that they can grow. I hope you all have fun with your experiment, and I'm excited to see what your results are. Bye!